Gene silencing. Gene silencing is a general term used to describe the regulation of gene expression in particular. This term refers to the ability of a cell to prevent the expression of a certain gene. Gene silencing can occur during either transcription or translation and is often being increasingly used to produce therapeutics to combat cancer and diseases such as infectious diseases and neurodegenerative disorders. Gene silencing is often considered the same as gene knockout. When genes are silenced, their expression is reduced. In contrast, when genes are knocked out, they are completely erased from the organism's genomes and thus have no expression. Gene silencing is considered a gene knockdown mechanism since the methods used to silence genes such as RNAi, CRISPR, or siRNA generally reduce the expression of a gene by at least 70% but do not completely eliminate it. Methods using gene silencing are often considered better than gene knockout since they allow researchers to study essential genes that are equipped for the animal models to survive and cannot be removed. In addition, they provide a more complete view on the development of diseases since diseases are generally associated with the genes that have a reduced expression. Antisense RNA is a sense strand single stranded RNA that is complementary to a messenger RNA mRNA strand transcribed within a cell. Antisense RNA may be introduced into a cell to inhibit translation of a complementary mRNA by base pairing to it and physically obstructing the translation machinery. The first antisense therapeutic to reach the market is the drug Fomiversin, approved in 1998. Fomiversin is an antisense antiviral drug that was used in the treatment of cytomegalovirus retinitis. CMV in immunocompromised patients, including those with AIDS. It was administered via intraocular injection. Mipomersen was approved in the United States in 2013. Mipomersen is used to treat homozygous familial hypercholesterolemia and is administered by subcutaneous injection. There is a serious risk of liver damage from this drug. The effects of antisense RNA are related with the effects of RNA interference. RNA, the RNAi process found only in eukaryotes, is initiated by double-stranded RNA fragments, which may be created by the expression of an antisense RNA followed by the base pairing of the antisense strand to the target transcript. Double-stranded RNA may be created by other mechanisms, also including secondary RNA structure. The double-stranded RNA is cleaved into small fragments by dicer and then a single strand of the fragment is incorporated into the DNA-induced silencing complex or RISC so that the RISC may bind to and degrade the complementary mRNA target. Some genetically engineered transgenic plants that express antisense RNA do activate the RNAi pathway. This process resulted in differing magnitudes of gene silencing induced by the expression of antisense RNA. Well-known examples include the flavor saver tomato and two cultivars of ring spot resistant papaya. Flavor saver, flavor spelling is important, FLAVR saver, is a genetically modified tomato, was the first commercially grown genetically engineered food to be granted a license for human consumption. It was produced by the Californian company Calgene and submitted to the US Food and Drug Administration FDA in 1992. It was first sold in 1994. Through genetic engineering, Calgene company hoped to slow down the ripening process of the tomato and thus prevent it from softening while still allowing the tomato to retain its natural color and flavor. The tomato was made more resistant to rotting by adding an antisense gene which interfere with the production of the enzyme polygalactoronase. The enzyme normally degrades pectin in the cell walls and results in the softening of fruit, which makes them more susceptible to being damaged. Flavor saver This is genetically modified tomato was the first commercially grown genetically engineered food to be granted a license for human consumption. It was produced by the Californian company Calgene and submitted to the US Food and Drug Administration in 1992. It was first sold in 1994. Through genetic engineering, Calgene company hoped to slow down the ripening process of the tomato and thus prevent it from softening while still allowing the tomato to retain its natural color and flavor. The tomato was made more resistant to rotting by adding an antisense gene which interfere with the production of the enzyme polygalacturonase. Polygalacturonase. The enzyme normally degrades pectin in the cell walls and results in the softening of the fruit, which makes them more susceptible to being damaged by fungal infections. Some unmodified tomatoes are picked before fully ripened and are then artificially ripened using ethylene gas, which acts as a plant hormone. Picking the fruit while unripe allows for easier handling and extended shelf life. Flavor saver tomatoes, on the other hand, could be allowed to ripen on the vine without compromising their shelf life. The intended effect of slowing down the softening of flavor saver tomatoes would allow the vine ripe fruits to be harvested like green tomatoes without greater damage to the tomato itself. Types of gene silencing. First, transcriptional. Transcriptional gene silencing is a product of histone modifications and DNA methylation, adding of a methyl group at the 5' position of a cytosine that prevent the transcription of that area of our DNA. Post-transcriptional meiotic transcriptional gene silencing. Genomic imprinting is the epigenetic phenomenon by which certain genes are expressed in a parent of origin specific manner. Genomic imprinting. Genomic imprinting is an inheritance process independent of the classical Mendelian inheritance. It is an epigenetic process that involves DNA methylation and histone methylation without altering the genetic sequence. The epigenetic markers are established or imprinted in the germline, sperm or egg cells of the parents and are maintained through mitotic cell divisions in the somatic cells of an organism. If the allele inherited from the father is imprinted, it is thereby silenced and only the allele from the mother is expressed. If the allele from the mother is imprinted, then only the allele from the father is expressed. Forms of genomic imprinting have been demonstrated in fungi, plants, and animals. There are about 150 imprinted genes known in the mouse and about half that in humans. Appropriate imprinting of certain genes is important for normal development. Human diseases involving genetic imprinting, including Angelman syndrome and Prader-Willi syndrome. Paramutation is an interaction between two alleles at a single locus, where thereby one allele induces a heritable change in the other allele. The change may be in the pattern of DNA methylation or histone modification. The allele inducing the change is said to be paramutagenic, while the allele that has been epigenetically altered is termed paramutable. Then transposon silencing is a form of transcriptional gene silencing targeting transposons. Transcriptional silencing of transposons is crucial to the maintenance of a genome. The jumping of transposons generates genomic instability and can cause extremely deleterious mutations. The transposable element insertions have been linked to many diseases, including hemophilia, severe combined immunodeficiency, and predisposition to cancer. The silencing of transposons is therefore extremely critical in the germline in order to stop transposon mutations from developing and being passed on to the next generation. Additionally, these epigenetic defenses against transposons can be heritable. Studies in Drosophila, Arabidopsis, Thaliana, and mice all indicate that small interfering RNAs are responsible for transposon silencing. And position effect is the effect on the expression of a gene when it is located in a chromosome is changed often by translocation. This has been well described in Drosophila with respect to eye color and is known as position effect variegation (PEV). The phenotype is well characterized by unstable expression of a gene that results in the red eye coloration. In the mutant flies, the eyes typically have a molted or spotted appearance of white and red sections. These phenotypes are often due to chromosomal translocation such that the color gene is now close to a region of heterochromatin. The heterochromatin can spread stochastically randomly and switch off the color gene, resulting in a white-eyed sectors. Then RNA directed DNA methylation, RDDM, is an epigenetic process first discovered in plants. During RDDM, double-stranded RNAs, dsRNAs, are processed to 21 to 24 nucleotide small interfering RNAs, siRNA, and guide methylation of homologous DNA loci. In plants, dsRNAs may be generated from three sources: viral replication intermediates, products of endogenous RNA-directed RNA polymerase transcribed inverted repeats. Wait a minute, alignment is a bit confusing. Yeah. Viral replication intermediates, products of the endogenous RNA-directed RNA polymerase transcribed inverted repeats, transposable elements. Then next, transposable elements. Besides RNA molecules, a plethora of proteins are involved in the establishment of RDDM, like agronuts, DNA methyl transferase, chromatin remodeling complexes, and plant-specific polymerase and polymerase 5. All these act in concert to act methyl group at the 5' position of cyclosine. So, products of the endogenous RNA directed DNA polymerase transcribed inverted repeats, transposable elements. So, besides RNA molecules, a plethora of proteins are involved in the establishment of RDDM, like agronuts, DNA methyl transferase, chromatin remodeling complexes, and the plant-specific polymerase 4 and polymerase B. All these act in concert to add a methyl group at the 5' position of cytosines. In contrast to animal, cytosines at all sequence context may get de novo methylated in plants. RNA-induced transcriptional silencing. RNA-induced transcriptional silencing is carried out by a complex of proteins called the Ritz complex. The fission yeast of this complex contains agronot. The induction and spread of heterochromatic regions require the agronot and RDRP proteins. Indeed, deletion of these genes in the fission yeast P. pombe disrupts histone methylation and centromere formation, causing slow or stalled anaphase during cell division. In some cases, similar processes associated with the histone modification have been observed to transcriptionally upregulate gene. Meiotic gene silencing transfection is an epigenetic phenomenon that results from an interaction between an allele of one chromosome and the corresponding allele on the homologous chromosome. Transfection can lead to either gene activation or repression. It can also occur between non-allelic regions of the genome as well as regions of the genome that are not transcribed. Transfection is believed to occur through a variety of mechanisms. In some mechanisms, the enhancers of one allele activate the promoters of a paired second allele. Other mechanisms include pairing sensitive silencing and enhancer bypass of chromatin insulated through pairing mediated changes in gene structure. Then post-transcriptional gene silencing, or PTGS. RNA silencing or RNA interference refers to a family of gene silencing effects by which gene expression is negatively regulated by non-coding RNA such as microRNAs. RNA silencing may also be defined as sequence-specific regulation of gene expression triggered by double-stranded RNA, dsRNA. RNA silencing mechanisms are highly conserved in most eukaryotes. The most common and well-studied example is RNA interference, RNAi, in which endogenously expressed microRNAs or exogenously derived small interfering RNA. Post-transcriptional gene silencing. RNA silencing or RNA interference refers to a family of gene silencing effects by which gene expression is negatively regulated by non-coding RNA such as microRNA. RNA silencing may also be defined as sequence-specific regulation of gene expression triggered by double-stranded RNA, dsRNA. RNA silencing mechanisms are highly conserved in most eukaryotes. The most common and well-studied example is RNA interference, in which endogenous expressed microRNA or exogenously derived small interfering RNA, siRNA, induces the degradation of complementary messenger RNA. Other classes of small RNAs have been identified, including PV interacting RNA, piRNA, and the subspecies repeat associated small interfering RNA. The next race RNA, the nonsense mediated mRNA decay, NMD, is a surveillance pathway that exists in all eukaryotes. NMD or nonsense mediated mRNA decay. Its main function is to reduce errors in gene expression by eliminating mRNA transcripts that contain premature stop codons. So translation of these aberrant mRNAs could in some cases lead to deleterious gain of function or dominant negative activity of the resulting proteins. NMD was discovered when it was realized that cells often contain unexpected low concentrations of mRNAs that are transcribed from alleles carrying nonsense mutations. Nonsense mutations code for a stop codon. Converting an amino acid into a premature stop codon causes the protein to be shortened. An RNA interference. RNA is an RNA-dependent gene silencing process that is controlled by the RNA-induced silencing complex or RISC and is initiated by short double-stranded RNA molecules in a cell's cytoplasm where they interact with the catalytic RISC component agronode. It is a biological process in which RNA molecules inhibit gene expression of translation by neutralizing targeted mRNA molecule. It is also known as by other names including co-suppression, post-transcriptional gene silencing, PTGS, and quelling, QUELLING, and Few Fire and Craig C. Shello shared 2006 Nobel Prize in Physiology for Medicine for their work on RNA interference in the nematode worm Caenorhabditis elegans, published in 1998. Since the discovery of RNAi and its regulatory potentials, it has become evident that RNAi has immense potential in suppression of desired genes. RNA is now known as a precise, efficient, stable, and better than antisense technology for gene suppression. Two types of small ribonucleic acid molecules, microRNAs, miRNA, and small interfering RNA, siRNA, are central to RNA interference. RNAs are the direct products of genes, and these small RNAs can bind to other specific messenger RNA, mRNA molecules and either increase or decrease their activity. For example, by preventing an mRNA from being translated into a protein. RNA interference has an important role in defending cell against parasitic nucleotide sequence viruses and transposons. It also influences development. The RNAi pathway is found in many eukaryotes, including animals, and is initiated by the enzyme DISA, which cleaves long double-stranded RNA dsRNA molecules into short double-stranded fragments of about 20 nucleotide siRNAs. Each siRNA is unwound into two single-stranded RNAs, ssRNAs, the passenger strand and the guide strand. The passenger strand is degraded and the guide strand is incorporated into the RNA-induced silencing complex RISC. The most well-studied outcome is post-transcriptional gene silencing, which occurs when the guide strand pairs with a complementary sequence in the messenger RNA molecule and induces cleavage by agronode to AGO2, the catalytic component of the RISC complex. In some organisms, this process spreads cellular systemically despite the initially limited molar concentrations of siRNA. RNAi is a valuable research tool both in cell culture and in living organisms because synthetic dsRNA introduced into cells can selectively and robustly induce suppression of specific genes of interest. RNAi may be used to systematic shutdown of individual genes in the cell, which can help to identify its function. Variation among organisms. Organisms vary in their ability to take up foreign dsRNA and use it in the RNAi pathway. The effects of RNA interference can be both systemic and irritable in plants and C. elegans, although not in Drosophila or mammals. In plants, RNAi is thought to propagate by the transfer of siRNA between cells through plasmodesmata channels in the cell walls that enable communication and transport. A broad general distinction between plants and animals lies in the targeting of endogenously produced mRNA. In plants, mRNAs are usually complementary to their target genes and induce direct mRNA cleavage by risk, while animals, mRNA tend to be more divergent in sequence and induce translational repression. This translational effect may be produced by inhibiting the interaction of translation initiation factors with the messenger RNAs, polyadenine tail. Biological functions: immunity. RNase interference is a vital part of an immune response to virus and other foreign genetic materials, especially in plants, where it may also prevent the self-propagation of transposons. Plants such as Arabidopsis thaliana express multiple dicer homologs that are specialized to react differently when the plant is exposed to different viruses. Some plant genomes also express endogenous siRNA in response to infection by specific types of bacteria. These effects may be part of a generalized response to pathogens that downregulate any metabolic process in the host that aids the infection process. So, although animals generally express fewer variations of dicer enzyme than plants, RNAi in some animals produce an antiviral response. In both the juvenile and adult Drosophila, RNAi interference is important in antiviral innate immunity and is active against pathogens such as Drosophila X virus. A similar role in immunity may operate in C. elegans as Argonaute proteins are upregulated in response to various worms and overexpressed components of the RNAi pathway are resistant to viral infection. The role of RNA interference in mammal innate immunity is poorly understood and relatively little data is available. However, the existence of viruses that encode genes able to suppress the RNA response in mammalian cells may be evident in favor of the RNAi-dependent mammalian immune response. Although this hypothesis has been challenged as poorly sustained, provide evidence for the existence of a function-active antiviral RNAi pathway in mammalian cells. Other functions for RNAi in mammalian virus also exist, such as miRNA expressed by the herpes virus that may act as heterochromatic organization triggers to mediate viral latency. So, downregulation of genes, endogenously expressed miRNAs, including both intronic and intergenic miRNAs, are most important in translational repression and in the regulation of development, especially on the timing of morphogenesis and the maintenance of undifferentiated or incompletely differentiated cell types such as stem cells. The role of endogenously expressed miRNA in downregulating gene expression was first described in C. elegans in 1993. The plants. This function was discovered when the Joe microRNA of Arabidopsis was shown to be involved in the regulation of several genes that control plant shape. In plants, the majority of genes regulated by miRNAs are transcription factors. Thus, miRNA activity is particularly wide-ranging and regulates entire gene networks during development by modulating the expression of key regulatory genes, including transcription factors as well as F-box proteins. In many organisms, including humans, miRNAs are linked to the formation of tumors and dysregulation of the cell cycle. Although animals generally express fewer variants of the dicer enzyme than plants, RNAi in some animals produces an antiviral response. In both juvenile and adult Drosophila, RNA interference is important in antiviral innate immunity and in active against pathogens such as Drosophila X virus. A similar role in immunity may operate in C. elegans as Argonaute proteins are upregulated in response to various worms that overexpress components of the RNAi pathway are resistant to viral infection. The role of RNA interference in mammalian innate immunity is poorly understood and relatively little data is available. However, the existence of viruses that encode genes able to suppress the RNA response in mammalian cells may be evidence in favor of the RNAi-dependent mammalian immune response. Other functions of RNAi in mammalian virus also exist, such as miRNA expressed by the herpes virus that may act as heterochromatin organization triggers to mediate viral latency. Downregulation of genes. endogenously expressed miRNAs, including both um, intronic and intergenic microRNAs, are most important in translational repression and in the regulation of development, especially on the timing of morphogenesis and the maintenance of undifferentiated or incompletely differentiated cell types such as stem cells. The role of endogenously expressed miRNA in downregulating gene expression was first described in C. elegans in 1993. In plants, this function was diversified when the jaw microRNA of Arabidopsis was shown to be involved in the regulation of several genes that control plant shape. In plants, the majority of genes regulated by mRNA are transcription factors. This mRNA activity is particularly wide-ranging and regulates entire gene networking during development by modulating the expression of key regulatory genes, including transcriptional factors as well as F-box proteins. In many organisms, including humans, mRNAs are linked to the formation of tumors and dysregulation of the MR cell cycle. Here, mRNAs can function as both oncogenes and tumor suppressors. Evolution based on the phylogenetic analysis. The most recent common ancestor of all eukaryotes most likely already possessed an entirely early RNA interference pathway. This ancestor RNAi system probably continued at least one dicer-like protein, one argonaut, and one PV protein, and an RNA-dependent DNA polymerase that may also have played other cellular roles. The ancestral function of the RNA system is generally agreed to have been immune defense against exogenous genetic elements such as transposons and viral genomes. Related functions such as histone modification may have already been present in the ancestor of modern eukaryotes. Applications: gene knockdown. The RNA interference pathway is often exploited in experimental biology to study the function of genes in cell culture and vivo in model organisms. Double-stranded RNA is synthesized with the sequence complementary to a gene of interest and introduced into a cell or organism. Here it is reorganized as exogenous genetic material and activates the RNA pathway. Using this mechanism, researchers can cause a drastic decrease in the expression of a targeted gene. Studying the effects of this decrease can show the physiological role of the gene product. Since RNA may not totally abolish expression of the gene, this technique is sometimes referred to as a knockdown. To distinguish it from knockout, procedures in which expression of a gene is entirely eliminated. Extensive efforts in computational biology have been directed towards the design of successful dsRNA reagents that maximize gene knockdown but minimize off-target effects. Off-target effects arise when an introduced RNA has a base sequence which can pair with and thus reduce the expression of multiple genes. Such problems occur more frequently when the dsRNA contains repetitive sequences. It has been estimated from studying the genomes of humans, C. elegans and S. bombs, that about 10% of possible siRNA have substantial off-target effects. A multitude of software tools have been developed implementing algorithms for the design of general, mammalian-specific and virus-specific siRNA that are automatically checked for possible cross-reactivity. Depending on the organism and experimental system, the exogenous RNA may be a long-stranded designed to be cleaved by dicer, a short RNAs designed to serve as siRNA substrates. In most mammalian cells, shorter RNAs are used because long double-stranded RNA molecules induce the mammalian interferon response to form innate immunity that reacts non-specifically to foreign genetic material. Mouse oocytes and cells from early mouse embryos lack this reaction to exogenous dsRNA and are therefore a common model system for studying mammalian gene knockdown effect. Specialized laboratory techniques have also been developed to improve the utility of RNAi in mammalian systems by avoiding the direct introduction of siRNA, for example by stable transfection with a plasmid encoding the appropriate sequence from which siRNA can be transcribed, or by more elaborate lentiviral vector systems allowing the inducible activation or deactivation of transcription known as conditional RNAi. There are several opportunities for the application of RNAi in crop science for its improvement such as stress tolerance and enhanced nutritional level. Stress tolerance. So, RNAi in crop science for its improvement such as stress tolerance and enhanced nutritional level. RNAi will prove its potential for inhibition of photorespiration to enhance the productivity of C3 plants. Its knockdown technology may be useful in inducing early flowering, delayed ripening, delayed senescence, breaking dormancy, stress-free plants, overcoming self-sterility, etc. Functional genomics. Normal adult Drosophila fly, a common model organism used in RNA experiments. Most functional genomics applications of RNAi in animals have used C. elegans and Drosophila, and these are the common model organisms in which RNAi is most effective. C. elegans is particularly useful for RNAi research for two reasons. Firstly, the effects of gene silencing are generally heritable, and secondly, because delivery of the dsRNA is extremely simple through a mechanism whose details are poorly understood. Bacteria such as E. coli that carry the desired dsRNA can be fed to the worms and will transfer their RNA payload to the worm via the intestinal tract. This delivery of feeding is just as effective at inducing gene silencing as most costly and time-consuming delivery methods, such as soaking the worms in dsRNA solution and injecting dsRNA into the gonads. Although delivery is more difficult than most other organisms, efforts are also underway to undertake large-scale genomic screening applications in cell culture with mammalian cells. Approaches to the design of genomic-wide RNAi libraries can require more sophistication than the design of a single siRNA for a defined set of experimental conditions. Artificial neural networks are frequently used to design siRNA libraries and to predict their likely efficiency at gene knockdown. Mass genomic screening is widely seen as a promising method for genome annotation and has triggered the development of higher throughput screening methods based on microarray. However, the utility of these screens and the ability of techniques developed on model organisms to generalize to even closely related species has been questioned, for example from C. elegans to related parasitic nematodes. Functional genomics using RNAi is a particularly attractive technique for genomic mapping and annotation in plants because many plants are polyploid, which represent substantial challenges for more traditional genetic engineering methods. For example, RNAi has been successfully used for functional genomic studies in red wheat, which is hexaploid, as well as more common plant model systems. Medicine in adult C. elegans worm grown under RNA suppression of nuclear hormone receptor involved in disaccharase regulation. These worms have abnormal fatty acid metabolism but are viable and fertile. They may be possible to exploit RNA interference in therapy, although it is difficult to introduce long dsRNA strands into mammalian cells due to the interferon response. The use of short interfering RNA has been more successful. Among the first applications to reach clinical trials were in the treatment of macular degeneration and respiratory syncytial virus. RNA has also been shown to be effective in reversing induced liver failure in mouse models. Potential antiviral therapies include topical microbicide treatments which use RNA to treat infection. At Harvard Medical School in mice, so far by Herpes simplex virus type 2 and inhibition of viral gene expression in cancer cells, knockdown of host receptors and co-receptors for HIV, the silencing of hepatitis A and hepatitis B genes, silencing of influenza gene expression, and inhibition of measles viral replication. Potential treatments for neurodegenerative diseases have also been proposed, with particular attention to polyglutamine diseases such as Huntington's disease. RNA interference-based applications are being developed to target persistent HIV-1 infection. Viruses like HIV-1 are particularly difficult targets for RNA attack because they are escape-prone, which require combinatorial RNA strategies to prevent viral escape. Cancer RNA interference is also a promising way to treat cancers by silencing genes differently upregulated in tumor cells or genes involved in cell division. The key area of research in the use of RNAi for clinical applications is the development of a safe delivery method, which is it to date has involved mainly viral vector systems similar to those suggested for gene therapy. Due to safety concerns with viral vectors, non-viral delivery methods, typically employing lipid-based or polymer vector, also promising candidates. Computational modeling of non-viral siRNA delivery paired with in vitro and in vivo gene knockdown studies elucidated the temporal behavior of RNA in these systems. The model used an input bolus dose of siRNA and computationally and experimentally showed that knockdown duration was dependent mainly on the doubling time of the cells to which siRNA was delivered, while peak knockdown depended primarily on the delivered dose. Kinetic considerations of RNA are imperative to safe and effective dosing schedules as non-viral methods of inducing RNA continue to be developed. Despite the proliferation of promising cell culture studies for RNA-based drugs, some concern has been raised regarding the safety of RNA interference, especially the potential for off-target effects in which the gene with a coincidentally similar sequence to the targeted gene is also repressed. A computational genomic study eliminated the error rate of off-target interactions is about 10%. One major study of liver disease in mice reported that 23 out of 49 distinct RNA treatment protocols resulted in death. Researchers hypothesized this alarmingly high rate to be the result of oversaturation of the RNAi pathway due to the use of shRNAs that have to be processed in the nucleus and exported to the nucleoplasm using an active mechanism. Such considerations are under active investigation to reduce their impact on the potential therapeutic application. RNAi in vitro delivery to tissue still elucidates science, especially to tissue deep within the body. RNA delivery is only easily accessible to surface tissue such as the eye and the respiratory tract. In these instances, siRNA has been used to direct contact with the tissue for transport. The regulating RNAi successfully focused on target genes when delivering the siRNA to deep tissue. The siRNA must be protected from nucleases, but targeting specific areas become the main difficulty. This difficulty has been combated with high dosage levels of siRNA to ensure the tissues have been reached. However, in some cases, hepatotoxicity was reported. RNA interference has been used for applications in biotechnology and is leading commercialization in others. RNAi has developed many novel crops such as nicotine-free tobacco, decaffeinated coffee. That's amazing. Nutrient fortified and hypoallergenic crops. The genetically engineered Arctic apples are near close to receive US approval. The apples were produced by RNA suppression of polyphenol oxidase gene, making apple varieties that will not undergo browning after being sliced. That's amazing. Silenced apples are unable to convert chlorogenic acid into quinone product. RNAi has been used to genetically engineer plants to produce lower levels of natural plant toxins. Such techniques take advantage of the stable and heritable RNA phenotype in plant stocks. Cotton seeds are rich in dietary protein but naturally contain the toxic terpenoid product gossypol, making them unsuitable for human consumption. RNAi has been used to produce cotton stalks whose seeds contain reduced levels of delta cardenine synthetase, a key enzyme in gossypol production without affecting the enzyme's production in other parts of the plant, where gossypol is itself important in preventing damage from plant pests. Other crops. Another effort decreased the precursors of likely carcinogens in tobacco plants. Other plant traits that have been engineered in the laboratory include the production of non-narcotic natural products of the opium poppy and resistance to common plant viruses. Insecticide. RNAi is under development as an insecticide, employing multiple approaches including genetic engineering and topical applications. Cells in the midgut of some insects take up the dsRNA molecules in the process referred to as environmental RNAi. In some insects, the Effect is systemic as the signal spreads throughout the insect's body, referred to as a systemic RNAi. RNAi has varying effects in different species of Lepidoptera. Butterflies and oats, possibly because of their saliva and gut use, is better at breaking down RNA. The cotton bollworm, the beet arvy worm, and the Asiatic rice borer have so far not been proven susceptible to RNAi by feeding. Then transgenic plants. Transgenic crops have been made to express dsRNA, carefully chosen to silence crucial genes and target pests. These dsRNAs are designed to affect only insects that express specific gene sequences. As a proof of principle, in 2009, a study showed RNAs that could kill any one of the 4 fruit fly species while not harming the other 3. The International Potato Center in Lima, Peru, is looking for genes to target in the sweet potato weevil, a beetle whose larvae ravage sweet potatoes globally. Other researchers are trying to silence genes in ants, caterpillars, and pollen beetles. Monsanto will likely to be forced to market with a transgenic corn seed that expresses dsRNA based on gene SNF7 from the western corn rootworm, a beetle whose larvae annually cause $1 million in damage in the United States alone. Admittedly, dsRNA can be supplied without genetic engineering. One approach is to add them to irrigation water. The molecules are absorbed into the plant's vascular system and poison insects feeding on them. Another approach involves spraying dsRNA like a conventional pesticide. This would allow faster adaptation to resistance. Such approaches would require high-cost sources of dsRNAs that do not currently exist. In genome scale screening, genome scale RNAi research relies on high throughput screening HTS technology. RNAi HTS technology allows genome-wide loss of function screening and is broadly used in the identification of genes associated with specific phenotypes. This technology has been hailed as second genomic wave, following the first genomic wave of gene expression, microarray, and single nucleotide polymorphism discovery platforms. Petunia plants in which genes for pigmentation were silenced by RNAi. The left plant is wild type. The right plant contains transgenes that induce suppression of both transgene and endogenous gene expression, giving rise to the unpigmented white areas of the flower. The discovery of RNAi was preceded first by observations of transcriptional inhibition by antisense RNA expressed in transgenic plants. In an attempt to alter flower colors in petunias, researchers induced additional copies of a gene encoding chalcone synthesis, a key enzyme for flower pigmentation into petunia plants of normally pink or violet flower color. The overexpressed gene was expected to result in darker flowers, but instead produced less pigmented, fully or partially white flowers, indicating that the activity of chalcone synthesis had been substantially decreased. In fact, both the endogenous genes and the transgene were downregulated in the white flowers. Further investigation of the phenomenon in plants indicated that the downregulation was due to post-transcriptional inhibition. The downregulation was due to post-transcriptional inhibition of gene expression via an increased rate of mRNA degradation. This phenomenon was called co-suppression of gene expression, but the molecular mechanism remained unknown. Not long after, plant virologists working on improving plants' resistance to viral disease observed a similar unexpected phenomenon. While it was known that plants expressing virus-specific proteins showed enhanced tolerance to resistance to viral infection, it was not expected that plants carrying only short non-coding regions of viral RNA sequences would show similar levels of protection. Researchers believed that viral RNA produced by transgenes could also inhibit viral replication. The reverse experiment, in which short sequences of plant genes were introduced into viruses, showed that the targeted gene was suppressed in an infected plant. This phenomenon was labeled virus-induced gene silencing, and the set of such phenomena were collectively called post-transcriptional gene silencing. Craig C. Mallow and Andrew Fair's 1998 Nature paper reported a potent gene silencing effect after injecting double-stranded RNA into C. elegans. In investigating the regulation of muscle protein production, they observed that neither mRNA nor antisense RNA injections had an effect on protein production, but double-stranded RNA successfully silenced the targeted gene. As a result of this work, they coined the term RNA. This discovery represented the first identification of the causative agent for the phenomenon. Fire and Milo were awarded the Nobel Prize in Physiology and Medicine. Thank you for listening.